Okay, this is screencast for the tour of the cell PowerPoint. I'm gonna go ahead and share my iPad as usual. And I'm gonna, yeah, let's see, okay. And probably not gonna get it all in this one screencast, but I at least wanna do part of it. All right, so tour of the cell. I am going to introduce the cell project soon. I'm not sure about the due dates yet, but I will introduce that in class. So cells have a kind of range in size from about 1 to 100 micrometers in diameter. So 1 to 100 micrometers. And just so in case you didn't remember, a micrometer is 1 times 10 to the negative 6th meters, all right? The naked eye cannot see this small, so we have to use microscopes to visualize cells. So I just want to give you kind of, this shows you like a little bit of a range, whereas a frog cell, a frog egg, we can see by eye. It's one of the largest cells. And then but when we get into the kind of human cells, they are in this range of either a light microscope or an electron microscope, whereas frog eggs are still able to see with the naked eye. All right, so there's 3 main kinds of microscopy. We are going to be using, hopefully, 2 kinds in class. We will be using a dissecting microscope, which allows us to see kind of just a sample, but bigger. So for example, this is a little butterfly head actually, and we're talking about in the millimeter range, right? So that's our little scale. As we go up and we want to see individual cells, we can use a light microscope. So the light microscopes that we have in class go up to 400 times what you can see by eye, or in terms of magnification, 400 times. So you can start to see individual cells, but you can't see a lot of detail in those cells. Then we have the big bad boy called the electron microscope, where now you can see that that cell is much, much more zoomed in. And we're talking about, you know, on the individual cell, you can start to see more detail within a cell. Right, and just to give you a sense, I have done a lot of microscopy in my life, really hundreds and hundreds of hours. And I just want to show you a picture that I have taken where this is looking at a fly neuromuscular junction where the neuron and the muscles connect, where the neuron here is shown in green and where it is connecting to the muscle is shown here in pink. And I'm happy to talk about that kind of stuff with anybody, because it's really exciting. All right, so why are cells so small? We just said they're so small, they're beyond the naked eye, like you have to use a microscope, but why? Why are cells so small? Why can't we have giant cells? And it has to do with this key factor, which is the surface to area ratio, all right? The surface area to volume ratio of a cell. That is what is absolutely critical. If the surface area to volume ratio is too small, then you have a limited rate of exchange between the outer world and within the cell. And that can lead to death, all right? Oops, sorry, let me move my little head. And that can lead to dead. All right, so as the surface area, and you guys should think about this in terms of like just math, as you increase the surface area, increases by a factor of n to the second, right? As the volume increases, however, to n to the third, all right? So if we look at this small cell and we look at the surface area to volume, it is 6 to 1, which is a nice big high number. That's good, right? As cells get bigger, this would be a big cell, then you see our surface area to volume ratio is much smaller. And that is very bad because it limits the rate of exchange of nutrients and materials and wastes from inside the cell to the outside of the cell, right? So again, larger cells, so I don't know where to put my face, put it over here. Larger cells have less surface area to volume ratio. So again, cells need to exchange nutrients and wastes with their environment, right? So that means that no part of the cell can be too far away from the external environment. So imagine if you had some toxic waste build up in the cell and it was at the very center. It would take a very, 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 very long time for that waste to find its way out of the cell. However, if you were in a very small cell, nowhere in that surface is it very far from the middle of the cell, right? So this rate of exchange of nutrients and waste are really, really important. And that's going for inside the cell too, things coming inside the cell. So surface area to volume ratio, we want large, and that occurs in small. So that makes the cell size very small. All right, cells are fundamental to living systems of biology, just as the atom is to chemistry, right? They're the fundamental units of all living organisms. And the cell theory, hopefully I'll have time to play the song in class, there's a really cool song about this, is the following. One, all organisms are made of one or more cells, all right? All living organisms are made of one or more cells. The cell is the basic unit of life, and all cells arise from pre-existing cells. So you might say, well, wait a minute, how'd the first cell come about then? All right, and we're gonna talk a lot about how the first potential cell, the first protocell came to be in another section, right? And this is another fundamental thing that we're gonna come back to over and over. Cell structure is correlated to its cell function. And this is very similar to what I said before about proteins, right? Protein structure is also going to determine that protein's function. And the same thing for cells. The cell structure is going to help dictate what the function of that cell is. So the basic features of all cells, right? So every single cell on this planet has these four things. Number one, a plasma membrane, which is a phospholipid bilayer. Number two, an aqueous environment on the inside. It's called a cytosol or a cytoplasm. I use those words interchangeably, so you might hear me say both, but it's the watery environment inside of a cell. Then every cell has to have DNA, right? That is going to encode kind of instructions for how that cell is going to live and function. And then the fourth and the final thing are ribosomes, all right? So ribosomes are the little molecular machine that is going to help RNA and make it into protein, right? So we have many different types of cells. We have very simple bacteria. We have very complex animal cells, like, let's see, like the neuron that runs from our spinal cord all the way down to our big toe. We have some very, very complicated cells, right? All of them have plasma membrane, cytoplasm, DNA, ribosomes. Those are the features that all cells have to have in order to live. All right, so there's two main types of cells. They are called prokaryotic. This stands for before the nucleus, and eukaryotic, or having a true nucleus, right? So let's just write again. So all cells have those four items: plasma membrane, cytoplasm, DNA, and ribosomes, right? Then all of those cells can be classified as either prokaryotic or eukaryotic, right? Prokaryotic cells are like bacteria and archaea, which we'll talk about later. These are the simplest forms of life. They're generally very small. The DNA is not in a nucleus. It is just in this region of the cell called the nucleoid region, right? The DNA in these guys is a one single circular chromosome, one circular chromosome, right? And we'll talk about what those are in a little bit. And the cytoplasm has no membrane-bound organelles. Does it have ribosomes? Hell yeah, because all cells have ribosomes. But it's not going to have any of those complex things like mitochondria and endoplasmic reticulum and all those other cool organelles that you're going to learn about, right? So these are the simple ones. Then we have the eukaryotic cells. These are the complex cells. These form the bodies of animals, plants, fungi, protists, all of those things. All the multicellular organisms are going to have eukaryotic cells, right? These are generally much, much larger. We're talking about 10 to 100 microns or micrometers. The DNA is inside this membrane-bound organelle or membrane-bound nucleus. So here is the nucleus. And the chromosomes are actually linear. So small linear chromosomes. So here I'm showing you kind of 3 chromosomes, whereas over here in the prokaryotic cell, we have, again, a single circular chromosome. And the cytoplasm of eukaryotic cells has a lot of organelles, and we're going to learn about all of them coming up. Okay, so if you kind of drew a Venn diagram, and of course my Venn diagram didn't work on here, so I'm going to draw it down here. Plant and animal cells are going to be eukaryotic, all right? So in the eukaryotic cells, you can either be animal, or you can be a plant cell, right? They contain most of the same organelles. So here, I'm just going to draw a little Venn diagram. So this is showing the plant ones, and here we're showing the animal ones. In this region here in the center, you can see that this is the region that is overlapping. So all cells, again, have plasma membrane, and in eukaryotic cells, they are all going to have a mitochondria, a nucleus, a Golgi apparatus, and an endoplasmic reticulum. There's a lot of vocab in this section, so you're just going to have to start to get comfortable with all of these, and whatever that means to you. Make flashcards, say them a lot, tell your parents, endoplasmic reticulum, do all of those things. All right, so animal cells have some unique features, and those are centrioles and centrosomes, and we'll get to what those are, lysosomes, which is going to be like the cell stomach. It's going to have a lot of enzymes in it to help the cell eat, and then cilia and flagella, which you probably don't even think you know about, but if you have ever seen or heard about a sperm cell, you know that they kind of move around with this long whipping tail. That's a flagella. Cilia is just when you have a lot of this. All right, plant cells are going to have cell wall, which we already talked about, which is made of cellulose, that polysaccharide. It's going to have chloroplasts, which are necessary for doing photosynthesis. We're going to see that plants have a central vacuole, and plasmodesmata, which are just going to be holes in the cell wall, so they can still exchange materials. Okay, so the eukaryotic cells have a lot of internal membranes. All right. So we already talked about how the plasma membrane is on the outside of every single cell and creates a selectively permeable membrane where small nonpolar things such as gases, which are shown here, and the steroids can literally pass through that plasma membrane. However, large, polar, ionic, those cannot pass through. They get blocked. And so what we're going to see later is that they actually are going to use membrane proteins in order to get through the plasma membrane, right? However, on the inside of the cell, there is also a lot of membrane, all right? And when I mean membrane, I mean phospholipids, right? Creating these barriers. So we have the endomembrane system, where we have all of the organelles that have these kind of, where membrane kind of flows through a cell. And we'll draw a picture of that in class. So we have the nucleus, right? The nuclear envelope, which is shown here in this big purple blob. Then we have the ER, the endoplasmic reticulum, right? We have the Golgi apparatus. We have lysosomes, which again are going to act like the cell's stomach and going to help the cells eat food. We have vacuoles, which are important for taking things in and getting rid of things. And then we have vesicles. And all a vesicle is a little sac, a little sac of membrane, and it can hold various things, right? And so when I draw a vesicle like this, remember that this vesicle, if you zoom in, is actually going to be, dun, 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 a phospholipid bilayer, right? All the membrane that we just talked about are phospholipid bilayers. All right, and we are going to stop there, and we will, in class, draw out the endomembrane system. Hold on, let me see. Let me just, we're gonna go through one other thing. Let me come back, sorry. All right, the last thing we're gonna talk about in this particular PowerPoint is going to be the flow of information. So in eukaryotic cells, eukaryotic genetic information, and this actually eukaryotic and with prokaryotic cells, what you're going to see is that you have DNA to RNA to protein, right? So DNA to RNA to our little machines that do all the work, the proteins, right? In a eukaryotic cell, that DNA is in the nucleus, and it can never leave the nucleus. Can never, ever, ever leave the nucleus, right? DNA cannot leave the nucleus of eukaryotic cells. That DNA is transcribed, it's this process called transcription, into RNA, all right? So it goes from reading those bases A, T, C, and G to a different kind of A, U, C, G. Similar language, but a little different. And then that RNA is actually going to leave the nucleus, all right? DNA stuck in the nucleus, always. See over here in the nucleus. The RNA, once it's made from the DNA, once it's transcribed, can actually leave the nucleus into the cytoplasm, where it will find ribosomes. And the ribosomes will go through what is called translation, where they translate that RNA language into a completely different language of amino acids, right? So the DNA in prokaryotic cells is a single circle, one circular piece of DNA. In a eukaryotic cell, this DNA is organized into linear chromosomes, where each single chromosome here is one DNA molecule, and they have some associated proteins, right? If you zoom in on the nucleus, there's also deep down a region called the nucleolus. And the nucleolus is a site for rRNA synthesis, right? So rRNA synthesis. And we're going to learn more about rRNA in a little bit. Okay, so with that, I'm going to stop this one, and we'll start again on the rest in a little bit.